My name is Tyler Harman. I am a recent master's graduate at Grand Valley State University, more specifically working with the Annis Water Resource Institute. And today I'm going to present some of my thesis research that looks at ecological simulation of baseline immunity in Ostrangia poculata. So coral diseases have been suggested to contain a coagulation of different pathogens, all to enact a particular disease, such as white band, black band, and more currently, stony coral tissue loss disease. And these pathogens trigger immune responses within coral organisms by the interaction of PMAPs, or pathogen-associated molecular patterns that are associated with pathogenic bacteria, onto PRRs, or pattern recognition receptors that are associated with coral hosts. These are several types of receptors, such as toll receptors in the toll receptor pathway and lectins in the complement pathway, and they all trigger a distinct immune response. And the pathway we'll be focusing on today is the melanin synthesis pathway, and this has been identified in cnidarian immunity, but more specifically within coral immunity. Several studies have identified this particular immune pathway as being a powerful indicator of baseline immunity within a coral organism, where signaling proteins such as prophenyloxidase activate melanin, which provides antimicrobial defenses. However, in our particular instance, we aren't exactly sure what particular disease impacts Ostrangia. So in light of this, we can use a PMAP to simulate disease exposure without the guesswork of picking a particular disease. One can use commercial lipopolysaccharides, or LPS, to simulate disease exposure within a lab setting in order to determine pathway signaling. So this leads to my study species, which is the northern star coral, or Ostrangia poculata. This species of coral is classified as a temperate coral species and has a facultative symbiotic relationship with symbiotic algae. This means compared to tropical corals, which have an obligate relationship, this can live with and without algae, or in very small quantities. It can either be listed as symbiotic, which has algae in its tissues, or as aposymbiotic, which it's either devoid of algae or it has much smaller quantities. However, when it does contain symbiotic algae, its known exclusive algal symbiont is Breviolum physmophyllum. And with this coral being facultatively symbiotic, it doesn't fully rely on photosynthesis from these algae in order to sustain themselves. It utilizes heterotrophic feeding in order to function. And one of the unique properties of this coral is its wide distribution range. In the scientific literature, it spans as far north as Cape Cod in Massachusetts, down along the East Coast, and as far south as southern Florida and into the northern shores of the Gulf of Mexico. So why look at disease? There's currently no literature that focuses on disease infections with Ostrangia. So why would you look at this in the first place? Well, knowing the expansive spread of Ostrangia up and down the East Coast, these southern populations of Ostrangia could be exposed to numerous coral diseases that plague southern Florida. But in addition to this, a recent study done by Rotchen et al. In 2019 with microplastics could introduce disease in these northern populations of Ostrangia. This study reported the favorability of microplastics compared to typical organic matter that it would consume, and even more disturbingly, this species preferred microplastics that were covered in E. coli compared to quote-unquote clean plastics. Many studies have identified microplastics to have films of bacteria and other microbes, which could be harmful to Ostrangia. So really the overall goals of this study is to first and foremost understand the establishment of immunity within Ostrangia poculata, but also to understand the differences in its disease resilience between different things like symbiotic state, temperature, as well as season. And then some of the questions overall are, how will the exposure to LPS impact the concentrations of things like prophenyloxidase and melanin in symbiotic and aposymbiotic Ostrangia compared to ambient temperature treatments versus elevated temperature treatments? Collections of Ostrangia at a location within Fort Wetherill State Park in Rhode Island, both in the summer of 2019 and the winter of 2020. And we brought these corals back to our customer circulating aquarium system at the Annis Water Resource Institute that you can see off to the left. So we ultimately had two different treatments. We looked at thermal stress, looking at an ambient temperature treatment of 18 degrees Celsius and an elevated temperature treatment of 26 degrees Celsius. Right at the beginning of our exposure, we had placed the corals into these plastic containers that contained water with LPS concentrations of 5 micrograms per milliliter at an exposure rate of 12 hours. So after the exposure of LPS for that 12-hour period, we had followed similar protocols that were outlined in Palmer et al. 2011. So these fragments were flash frozen in liquid nitrogen, as you can see off to the right, and they were stored in a -80 freezer until further processing. So once we were able to process these coral tissues, they were air blasted from the skeletons and processed in a buffer solution and ground with a pestle and mortar. And so to analyze the melanin within these coral tissues, we took 3 replicates of this tissue slurry and added 10 molar of sodium hydroxide to these samples and allowed them to dissolve overnight. And so the next morning, we added samples to a 96 well microplate and analyzed each of them at 410 nanometers, along with a standard curve of commercial melanin in order to determine the concentration. In order to determine the amount of melanin based on the size of each fragment, we actually took surface areas of each fragment that we're calculating using a modified paraffin wax technique that's outlined in Holmes et al. 2008. This gave us the milligrams of melanin per square centimeter of each fragment that was analyzed. And for the prophenyl oxidase analysis, we subsequently, after the aliquots of the melanin analysis, took these samples and flash froze them once again and extracted the proteins out of each sample. And after this extraction, we took 3 aliquots and stored them in a -80 freezer until further analysis. These 3 replicates were then added to 96 well microplates and had additions of trypsin, L-DOPA, and a PBS solution to each well. And these plates were analyzed at 490 nanometers over a period of 15 minutes to determine the change in absorbance over time, and these data were normalized to milligrams of protein that were run through Bradford protein assays. So our statistical analyses involved looking at Shapiro-Wilk tests to determine normality, and after a log transformation, all of our data went through a 3-way ANOVA, looking at things like season, symbiotic state, and temperature treatment, and utilized a Tukey's HSD post-hoc test to look at targeted comparisons. So our very brief results from these data sets show that symbiotic state actually had a significant effect on melanin concentrations, depicted by this bold p-value that we can see off to the table on the right. And upon further analysis with the Tukey's post-hoc test, we found that when comparing the interaction term between temperature treatments and season, we depicted a significant difference between symbiotic and aposymbiotic fragments in the winter collections, which show a much higher melanin concentration in the symbiotic compared to our aposymbiotic fragments. And regarding the interaction term between temperature treatments and symbiotic state, we had found that within ambient temperature treatments, symbiotic also had much higher melanin concentrations compared to the aposymbiotic ones. And with our prophenyl oxidase analysis, we unfortunately did not find any significant influence from temperature treatments, season, or symbiotic state. And so by analyzing our results, we found a lack of differences in prophenyl oxidase between our symbiotic and aposymbiotic fragments. And with the low melanization or low concentrations of this signaling protein, this could mean that the species is considered disease susceptible, as this immune response wasn't upregulated across different temperature treatments. And this low melanization could identify that other immunity parameters may have priority over the melanin synthesis pathway, as this was seen similarly in Palmer et al. 2011, which could suggest that pathways such as the complement pathway have a higher priority. And in addition to this, the lack of differences in prophenyl oxidase could also suggest the stability in environmental conditions that were not severe enough to really see change. Now, analyzing the melanin concentration data, we do see a significant influence of melanin concentrations from symbiotic state, where symbiotic fragments had higher amounts of melanin compared to our aposymbiotic ones. This could suggest a few hypotheses. First, as addressed earlier, other parameters within a Strangia's immune responses may have a priority over the melanin synthesis pathway, and we suggest that there is an influence in the complement pathway in the species over the melanin synthesis pathway, as other studies have identified the complement pathway as being involved in both immunity as well as symbiotic interactions with mutualistic coral and algae. And some studies have even gone further to address the complement pathway within coral immunity by subjecting algal cultures to concentrations of LPS and reporting their responses to PMAPs. And a review paper by Mansfield and Gilmore actually had identified that when symbiotic interactions in a coral take place, there are significant downregulations that occur within the complement pathway immune responses, suggesting that symbiotic interactions can essentially block this immune pathway from activating when exposed to disease. So this suggests that based on our data, the downregulations in the complement pathway occurred within symbiotic Ostrangia, and the melanin synthesis pathway could be considered a backup immune response when symbiotic interactions take place. As with the aposymbiotic Ostrangia, this could impose that the complement pathway was the preferred immune response given the low concentrations of the melanin that were found within our data. And lastly, our melanin analysis showed this influence of symbiotic state within our winter collections. This was a little harder to identify the potential avenues for interpretation, but the one thing to note is the importance of the surface mucus layer within coral immunity. The surface mucus layer actually contains helpful bacteria as well as antioxidant properties in order to mitigate pathogenic infections. And Sharp et al. In 2017 have identified that within the coral microbiome, which does include the surface mucus layer, changes did occur on a seasonal basis. So particularly within the winter, there were significant differences in the microbiome between symbiotic state, suggesting that our aposymbiotic Ostrangia had a much different microbiome makeup compared to our symbiotic Ostrangia. We suggest that the difference here in the surface mucus layer could have influenced the differences that were found within melanin concentrations. So regarding our disease impacts, we currently hypothesize that this species is susceptible to disease. However, there are many other parameters that need to be researched in the near future, such as the complement pathway, in order to really fully understand this species' overall immunity. And with that, I will take any questions and comments. You can contact me by my email on the screen there. And thank you so much for tuning in.